fingernail, you can see the sloughing of skin from your body, stratified squamous epithelial tissue that is constantly being sloughed off of your body and out into the surrounding environment. In other words, you're constantly shedding cells from this tissue into the surrounding environment. It's a continual process. Okay, is everybody good? Good on this? See what kind of advantages a microscope gives you as far as being able to look at things in a lot more detail. Now let me say that with this project that Drew and I are doing, a lot of times some of the adult insects we work with are extremely small. So in order to be able to distinguish between a lot of these really small insects as well as larger ones, you need that level of detail enlarged greatly, and we have to use microscopes to do that. And when it comes down to the level of identifying things to species, we oftentimes will have to dissect the specimen and dissect reproductive organs out of those specimens in order to determine what species they belong to. So in some instances, it may entail dissecting out genitalia, reproductive organs, out of an adult specimen that may be no larger than 1/2 millimeter in length. So you think about the specimen is 1/2 millimeter, and then we're going to be dissecting internal reproductive organs out of that little tiny specimen. So as Drew has learned with working with a lot of these tiny things, at high magnification, when you are working with little tools, because your fingers are too big to manipulate these little things under the microscope like you're doing this stuff now, you've got to use little micro tools. And any slight bit of movement by you with those micro tools is huge at high microscopy. I mean, the slightest little move or little bit of shake in yourself is like this at high magnification. So you learn to become quite steady in doing this job in order to achieve the results that you're trying to get. Let me mention one other thing about these scopes. As I said, the range of microscopy, as it sits, as you have right now, ranges from a low of 8 times magnification to a high of 32 times magnification. Well, we have something we have here to help with that. And that is this piece. What this is what we call a front lens, or a doubler lens. It's a 2x lens. This thing is a huge, thick piece of very precisely tooled glass that we screw on to the bottom side. And when that lens is screwed into place, now your magnification, this magnification range, is doubled. It goes from a minimum of 16x to 64x. So this is very, very helpful when it comes to working with some of these specimens that are really, really, really tiny. Because even the way your scopes are set up right now, 32x, it's going to be sufficient to see some of the things, some of the details that we need to see. So we've got doubling lenses like this that we'll use with these microscopes in some of our labs over here. Think about, just a lens like that, and it gives us double the magnification. But you think about, well, what kind of cost does something like this add to the price of the microscope? I believe these were anywhere, I think they were either $600 or $800 just for this lens. So microscopy equipment is cheap. All right, any questions about the use of the microscope, proper use of the microscope? Okay, if you have any money that you were looking at, you can go ahead and pass that in to me now. Okay, so what we're going to do, the next part of this workshop, I got to go back just a second.
Well, that was yesterday.
So it was yesterday.
Because Thursday is like my lab day, so I'm really I'm walking on Thursday.
Oh, yeah.
I think you could just like walk in there and sign a piece of paper and then well, not now because we're on video.
Okay, we're going to do a little simple exercise here involving insect identification. So what I'm giving you here is a key to insect orders. And in biology, had biology courses in the past, probably at some point were introduced to Linnaean taxonomy, which has to do with ranking identification levels for living things. Is that scheme of kingdom, phylum, class, order, family, genus, species. Everybody knows that, right? Okay, so what you're going to be doing is you're going to be looking at identifying insects to the order of to the level of order, the rank of order. So, in this project that Drew and I are working on, this is where the title of this workshop comes from. You have an unknown specimen, you put it underneath your microscope. The question is, who are you? What species do you belong to? So you have to go through a process of identification to make that determination about that data point that you're trying to determine what species it is. Now, there's a lot that goes into determining species, so you're going to get a small taste of kind of things that lead to that by identifying some insects to the level of order. So in order to do this, in order to do a lot of the identification processes, we use what are called dichotomous keys, and that's what you hold in your hand is a dichotomous key. So what a dichotomous key is about is where you have an unknown specimen and you're wanting to determine something about it, in this case being what order does it belong to. You go through a series of choices where there are two choices. So look on that key that you've got there. Notice number one. You have one A and one B, two statements there. That paired statements of number one, that's what we call a couplet. So the dichotomous key is a series of couplets. So when you read that couplet number one, you all start at the beginning. You look at your unknown specimen and you look at those two statements. Insects with wings or insect without wings. So you look at your specimen in question and see, does it have wings or does it not have wings? Let's say it doesn't have wings. You would then choose the B part of that couplet and you look out to the right and it says 28. What that number is, that tells you the next couplet to go to in this dichotomous key. So you go to couplet number 28 and you should find another second series of questions there, or statements. And at 28, the couplet is, are antennae present or antennae absent? And if the antennae are absent, you go to 39. If the antennae are present, you go to 29. Okay, so let's go with 29. Let's say that there are antennae. Then you go to 29, asks you, are the mouth parts usually withdrawn or enclosed in the head and not apparent, or are the mouth parts usually distinctly apparent? Well, let's say that they're distinctly apparent. We go to 31. In 31, the question is does that long tail-like appendage that's absent, or is there tail-like appendages present? Let's say there were tail-like appendages present. If you chose that, then you see over to the right, instead of another couplet to go to, you see a name there, Thysanura. If you have made the correct choices at each of the couplets, your unknown specimen you're looking at is a type of silverfish or firebrand, which is what we call the Thysanuras. insects that belong to the order Thysanura. And you would have correctly identified that specimen to order. Okay? So I don't expect you all to know insect morphology as far as, body parts and all that. But that's what these little pictures and things you see are, to kind of give you some clues, and things are pointed out as to what's meant by some of these statements that you find within the covenants. But also, if you turn over to the very back of the back page, there are some basic insect parts that are labeled there as far as the overall insect body, parts of the legs, and also parts of the head. Because these are all parts that are involved in these couplets that have been created to use in this dichotomous key. Okay? So, what I'm going to have you do is I have preserved insect specimens here for you to work with and to try and identify to order. So first of all, to give you something to put these specimens in, we'll let you reach in and grab a block of styrofoam here. Grab a block. I'll just pass this down, let y'all pass it down, and you can grab one out, pass it on. Okay, everybody get one? Why the styrofoam? Well, the way that the particular specimens, insect specimens, are preserved that you're going to be looking at are specimens that have been pinned to be preserved. So to give you an idea of what we're talking about, here are some pinned insect specimens. Okay, these pinned insect specimens are very dry, they're very brittle. They have sometimes very delicate little parts like antennae and leg parts that break very easily if they're jarred around too much or if they're bumped against something accidentally. So you need to handle these pinned specimens very carefully, but you're going to stick the pinned specimen. Like this dragonfly that I've got here, the pinned specimens, on the top of the pin that's been passed through the specimen, there's a little tiny ball that is just perfect for you to grab a hold of with the tips of your fingers. You grab a hold of that, that's basically the handle for you to handle this insect without damaging it. But you're going to take that and you're going to push the end of the pin down into the styrofoam, and then that acts as a holder for you to look at that pinned specimen underneath your microscope. Now going back to the work that Drew and I are doing, all the insects we're working with are ones that have come out of water. And instead of being pinned, those specimens have been preserved in ethanol, in ethyl alcohol. So we actually work with and look at and identify the specimens while they're submerged in alcohol. But we're not going to mess with any of that. You're going to do this simple exercise using pinned specimens. Okay? So I've got a whole host of preserved specimens for y'all to look at. You don't have to do just one. You can do many of these if you like. But these boxes that I have up here on the table have a variety of insects in them. As you can see, most of these insects that are in these boxes are local insects, ones that have been collected from here in north central Texas. But I can't stress enough how brittle these are, and you need to handle them very, very carefully as you look at them, as you turn and manipulate them underneath the microscope. One of the things with that block that you got there in front of you, not only can you use it to stick a specimen in the top to get a top view of that specimen, but say you want to look over on the side of the specimen. You can take that thing, stick it in.